for having me come speak today. I kind of will slant much more towards the sustainability side than the welfare side. But yeah, as you will see near the end of my talk, we are integrating some welfare aspects into the research we're doing, specifically with Dr. Regmi. He's kind of who invited me to give this talk today. As you said, I'm a microbial ecologist, so I'm kind of interested in how the environment shapes these bugs, specifically the bugs that can get us sick. When I first started out looking at poultry, it started in the conventional system, but then when I saw some of these pastured poultry farms around in my part of the world in Georgia, I just thought there was a lot of research opportunities to see how the environment and different aspects of the environment affect these pathogens and affects the safety of these products. That may not be a large component of the overall poultry market, but a very important one, especially in local and regional communities. That's kind of what I'll give my little spiel about today. Starting in about 2014 or so, we started a pastured poultry project in my lab where we followed over 4 years, we followed 42 pastured poultry flocks across 11 different farms in the southeast region. It was not just within my state, but it was also based on whoever was willing to allow me on their farms to do the sampling. We able to convince 11 farms to help us out with that. A little bit schematic on the right-hand side of the screen. What we did is we followed them throughout we did a farm to fork perspective. So we followed them when they were on pasture through the processing and onto the final product. And then my idea was basically to sample as much as we can collect as much data as we could to see who knows what's affecting these pathogens. In order to figure that out, we needed to collect as much data as we could. So the data included things like management data. So what we had were questionnaires for all of our farmers. It was about a 12, 15-page questionnaire talking about the management of the birds, the management of other animals on the farms, agricultural animals, things like the feed, where they get the feed from, where they get the chicks from, how often they move the chicks, where their water comes from, how they process the chicks, things like that. We collected physiochemical data from the pre-harvest samples, so that would be soil and feces, so things like pH, moisture, nutrient content. We did collect meteorological data, but we did it retrospectively. So after we sampled, we went by zip code and collected different meteorological data for that zip code during the times when we were sampling. And then we did a variety of biological data. So for that, mainly we did it in two ways, but one was culture-based. We looked at the major pathogens, human pathogens, so Salmonella, Listeria, Campylobacter, and E. coli. And then we would also did some molecular analysis looking at microbiome, so whole microbial populations. For today's talk, I'm going to focus mostly on the Salmonella work that we did, but we published tons of papers on this stuff so far. So if you have questions about the other stuff, I can direct you to that and we can talk more about that. But in the interest of time, we will just focus on the Salmonella for this talk. In terms of the Salmonella, what did we find? The table here on the left side of the screen, you've got the different types of samples on the top half of the graph and then the different farms. We just blind labeled them, so farms A through M, just to see the diversity and the ecology. So the pre-harvest setting, anywhere, 12 to 15% of the samples were positive in the pre-harvest setting for Salmonella. In the post-harvest, it was a little bit higher. You can see they're only in the processing whole carcass rinse. So that was the whole carcass rinse that was after processing, but before it was stored for however long it was for that farmer to get the product to the final consumer. It was a little bit higher in that processing whole carcass rinse, but it dropped to the normal level with everything else by the final product. We also observed that when we look in the bottom half of that table, between the farms, there is a very high variation farm to farm in terms of the Salmonella prevalence that we would find. Some would have a relatively decent amount, some would have none at all. And unfortunately, this was all based on farmer participation. We never asked the farmers to do anything specifically for us. We just followed whatever they were doing. So some farms we got a lot of samples from and followed multiple flocks from. Some we couldn't do that. It was variable in that regard. But we did see a bunch of differences. But just looking to see if there was salmonella there or not, to me, isn't sufficient. It's more important to look which salmonella are there, because not all salmonella are created equal, and some are more important in terms of public health and food safety issues than others. Um, let's see. There we go. So the next thing we did is for all the isolates we got, we went and serotyped them to identify who they were, not just if they were salmonella. So what we found there that Salmonella Kentucky was by far the most prevalent serotype, about three-quarters of all the isolates we recovered. Kentucky is within the United States is a serotype that has never been known to or shown to create human illness. So in that regard, it's I don't know if it's you can say good, but it was filling the salmonella niche of a serotype that does not known to cause disease. And then if we looked in terms of the ones in the red box here, so these are serotypes that the CDC in their top 30 serotypes known to cause human illness. So yes, we did have a good number of them in terms of the number of different serotypes, but it only represented about 16% of all the isolates recovered over 4 years. So a relatively small amount. And I would say that these, from the last slide and this slide, these are completely in line with what you would find from a commercial facility. So I don't think, even though they have access to a lot more the environment, a lot more environmental drivers and stressors and things like that compared to conventional, we didn't see a vast difference in terms of salmonella. But since we saw prevalence was very farm dependent, we next wondered, was the diversity of the serotypes we discovered farm dependent? And so what we found is when we looked over the different sample types, so not just the farms at this point, the sample types, the different colors are different serotypes. Yes, you see some differences there, but largely, can I go back? Largely, they were relatively stable over the sample type. So what we found in one sample type was largely what we found in another sample type. But when we looked at across the different farms, we saw very distinct populations of salmonella on different farms, and you can see the different colors there. So it appears that the farm of origin was a much more significant factor in determining the diversity of salmonella than the type of sample on a farm that we were recovering it from. And while I'm just talking about the salmonella data here, this is something that we found occurred in Campylobacter and Listeria and E. coli. All of them the farm location, farm to farm, there is very distinct differences. And especially in a pastured poultry setting, the methods and the management practice were very diverse, so it probably relates back to management practice in some way. These were all farms that never used any antibiotics in their feed or as medications at any point. But we still were interested to see what kind of antibiotic resistances the salmonella had, because these would just be normal, natural levels of background baseline resistance. And so in the absence of any use, about 27% of all the isolates we recovered were what we called multi-drug resistant. And that means they were resistant to 3 or more of the antibiotics we tested them against. And I think the panel had 8 to 9 antibiotics on it. And aside from the Zika, the rates were relatively similar across the sample types. But what I'll show you in the next few slides is some farms exhibited very unique antimicrobial resistance profiles, which we found interesting. So here is just a subset of some of the farms that we saw. And so these are different. Each column here is a different antibiotic resistance. And the blue area of the bar indicates how many isolates recovered from that farm were resistant to that the antibiotic. And so for like farms B and E and M and I, they were largely sensitive to all of them, except sometimes for the two most common, the streptomycin and tetracycline over here. But what you see is from farms A and H, you see these first 5 antibiotics, they have greater than 50% resistance to. And the interesting thing there is that Farm A, for the first 2 years of our study, Farm A was at a certain farm. And then at one point, they had to move their farm due to leasing issues to another farm. And then in the third year, they were at a second farm. And then they moved again to one that they could purchase themselves to a third farm. And the third farm, when we followed them, that was what we called Farm H. So farms A and H are the same infrastructure, the same farmer, the same equipment, but just different physical locations. Excuse me. And so you saw this unique pattern of resistances followed it. So there is a certain plasmids that salmonella can have, and it's potentially indicates that that plasmid was somewhere on the infrastructure, either on the equipment or on the housing or something that when they moved it, it went along with it. So even when they were on different physical land, and I will say that A and H, they were in different states at this point, and one was up in more in the mountains, so it's very different regions and climates. But that held true. So there definitely seems to be the infrastructure of the farm seems to have some potential for carryover between flocks. So that was just pure based on the microbiology. Luckily, I've had the opportunity to work with a couple different groups at different universities to do some modeling of this data that we have. So the first thing I'll talk about is with Dr. Misra at UGA. He's in the food science department. So basically, like I said, we have all of this data, the management data from the questionnaire, the meteorological data from the zip code, the retrospective review, and we have all this data about the salmonella that we found in the different samples in the pre-harvest and the post-harvest setting. So that's a lot of data, and sometimes a lot of these relationships aren't nice and linear. Like this one variable is the only one that's important. No, usually it's a combination of many variables working together or against each other that have an effect. And using traditional tools, that was very hard to understand, but now we're using some of these modeling tools. So what we've done is with his group, we've done it's called random forest modeling. But basically what it does is it takes all this data and combines it together and combines all the variables in thousands and thousands and thousands of different ways to figure out in which combination of and ranks the importance of the variables, which ones are the most important ones based on all these thousands of models that result in a positive salmonella sample versus a negative salmonella sample. Excuse me, I've been ill the last couple of days. So what this is identifies in a different way the variables that have greatest importance to salmonella prevalence on farm. So when we look at some meteorological factors, so this is a graph of the relative importance of these different variables. So things such as the maximum gust speed, average temperature, average humidity, those are the ones that seem to be of greatest importance to whether a sample was salmonella positive or not. And what we were able to find is that this graph here for the wind gusts, when wind gusts were less than 10 meters per second, the predicted probability of salmonella was lower. But as the winds got higher, greater than 11, then it increased. And there is some evidence that salmonella can survive as a bioaerosol on dust and everything. So the windier got, it may indicate that it could travel. And the more the salmonella traveled and was positive, the higher the prevalence was. In terms of average temperature, the week before sampling, when they were less than 28 degrees Celsius, it had a lower prevalence, but once it creeped up to 30 and above, so as it got in the warmer season, then you saw salmonella levels increase. And this is just telling you, this is just a metric for showing you how good the model is. A perfect model would have an AUC of 1, and we had one 88%, so that's quite a good model. So basically what that means is it's very accurately indicating which variables are important. Excuse me. In terms of management factor, things like years farming and the type of pasture feed seem to be most important. So for the years farming, the longer they farm, the higher the probability of salmonella detection is. That could, again, it could be a combination of the infrastructure having an importance in terms of carryover between flocks, and also indicates the need of a good biosecurity cleaning regimen to keep that influence lower. And then what we found with the feed was that corn-based feed seemed to have a higher probability of salmonella versus ones that are more based on wheat and oat. In terms of the post-harvest, so this is for what we found on the carcass rinses. Again, feed, corn-based feed, once again, showed to have the greatest influence on probability of finding salmonella. The non-corn feed was lower. And then it looked like the flock in ages, and this is a weird thing, but if you had a very short grown flock, less than 10 weeks, you had a high amount. For some reason, if it was in the sweet spot of 10 to 12 weeks, it would drop, but then it would go back up to the same level after 12 weeks. I'm not sure exactly what that means, but if that does hold true in future studies, that could indicate for a farmer a good time period for which to raise the flock in order to minimize their risk of salmonella being present in the final product. And then the next thing we did is we went a little bit deeper with the modeling with a group from Mississippi State, and that doctors Nanduri and Ram Kumar. And so up to that point, we did all the predictions on a single model. But again, these are complex systems with lots of variables. So what they did was they did an ensemble approach, and all the information, I'm putting it there just to show you what we use. But the important thing is they did a bunch of modeling on 5 different models with the thought of if many of the different models showed the same variables as important, that's an increased likelihood that in the real world, those are important variables. If only one model showed it was important and the other 4 didn't, maybe it's just a weird bias of that singular model. But if we used 5 and on most of those 5 it showed a feature was important, then that's something that probably we should investigate further. So just in terms of what we looked at, again, we looked at the management features again, but this time we delved deeper into some of the physiochemical parameters of the feces in the soil from which we isolated the salmonella. And you can see the list here on the right side of this graph. So really trying to understand what in the matrix that these salmonella are growing in are potentially allowing it to survive. So when we looked at these critical farm values, again, for the feces on the top and the soil on the bottom, the next column shows the 5 different models. And then the rest of the table shows the rankings that the different parameters on the top here had. So, for example, magnesium was the 5th most important one in this model, the 3rd in this model, the 3rd in those models. And you can see, and basically what we did is we put boxes around the ones that were found to be important in 3 or more models. And we estimated that if it was important in 3 out of the 5 models or more, then those are important things that we should investigate. And the power of this kind of analysis versus the predictive modeling is what we were able to do is, because this is quantitative data for a lot of this, we have discrete values for exactly what the potassium levels are, for what the copper levels are and things like that. We can make recommendations to reduce prevalence based on this data. So what we found is, like for fecal in the feces, if you want to reduce salmonella in the feces, you want to have less than 5,000 ppm potassium in the feces itself. You don't want to have GMO-free pasture brood feed. Things like that. So this allows us to give very specific recommendations to farmers that some of which they may be able to control, some they can't. For instance, like the soil, yes, you can control the number of birds potentially on your farm. You can control average number of birds. You can control the flock size. You can control some of the nutrient components in the soil potentially. And also within this data, what we're able to find is that we can make different recommendations, because within our data set or within our farms, some of them were what we considered hobby farms. So they were doing 1, at most 2 flocks a year. And then the professional ones were doing 3 or more flocks a year at much higher numbers. And what we found is we can make distinct recommendations for those 2 sizes, because within the pastured poultry community, at least in my experience, and maybe Mike Badger in the 3rd talk can speak more to this, there is a range, and we did run into a lot that were in the much smaller scale, and then and much fewer at the higher what we call professional scale. So it would be good to give recommendations to the two different groups based on that size, because it's not a one size fits all model. Um, and one last thing in terms of MDR, we wanted to see if any of those things had an effect on the multi-drug resistance rate of the salmonella that we recovered. And again, I won't go through the numbers, but we were able to find that certain chemicals were indicative of MDR rate within the salmonella positive samples. It seems things like magnesium and electrical conductivity of the samples affected the MDR rate. And other things such as phosphorus, manganese, and sodium also affected that rate. So you could see these are things that not only affected the prevalence of salmonella, but also the rate at which the salmonella we recovered were multi-drug resistant. So some general conclusions from that data there is that pasture poultry farmers, they similar prevalence rates to commercial farms, and about 85% of the isolates were not serotypes of the greatest concern to human health. And it seemed of all the things we looked at, the farm environment was really the driving factor for the diversity and the serotypes and the phenotypes that we recovered. And the modeling, the machine learning algorithms helped us identify these management or environmental factors that are predictive of salmonella prevalence and MDR rates. That's not to say that these are universally true. These could be specific to just this area of the country that we got our farms from. But it does give us things to start to investigate further, to look further at, and develop new studies to see if those things hold true on larger data sets and more diverse data sets. So that's some of the stuff that we've been doing. I just want to finish up my part of the talk here to talk about where we're going in the future and where we went last year and where we're going in the future. So like I said, all of these were farm studies. So I just went to the farmer and said, you do whatever you're going to do, just please let me come in and sample certain things. And, for the most part, that's how that went. Every once in a while, some farmers are like, well, if you want to do something, let me know. I'm willing to do it. In that case, we did a couple small little things. But for the most part, I couldn't control what was going on. And scientifically, that's difficult to not have control over the studies. So what we're doing now is we're translating these farm studies to some more controlled field trials. So one of the first things we did this past year, we looked at compared the two different types of breeds that you see in pastured poultry. So usually there's a fast-growing breed, so it's some type of Cornish cross, versus a slow-growing breed, which is some kind of ranger, a Freedom Ranger, Red Ranger. They have a variety of different names, so the Cornish cross comes from a more commercial line, whereas the Ranger one is not a commercial line at all. So what we did this past summer is we grew them side by side. The dates aren't important here, but so basically we grew them same brood period. We brooded them together until day 18. We put them on pasture, and then grew them out until they're about 3.5 kilos of growth. So for the fast growing Cornish crosses, that was till about day 56. And to the slow growing Freedom Rangers, that was to day 77. And here's just some pictures. I will say that this was, we have these automated coops, which we have a picture of in the bottom right here. This is through a company called Chictopia that built them for us. So basically, these are automated coops. They have wheels, they have solar panels on top, and they have water and feeder water lines with a big water reservoir in the back and a pump that the solar power powers to push water through these systems. And then we have a hopper with a corkscrew driven feed line that the solar power also drives. And so those are automatically run throughout the day. And then we can move them space to space after that. So they're contained. These are more controlled systems that we could do some things with for the scientifically. I don't know how feasible these may be for a normal farmer every day, but in terms of scientifically, that was helpful for us. And here's some pictures of these coops on this field trial. So here you can see them growing out on the pasture. So we had 4 coops. 2 of the coops were the Freedom Rangers, and 2 of the coops were the Cornish crosses. We started with about 150 birds in each coop. But then we did end up destructively sampling. We did necropsies at different times throughout to get different samples. But anyhow, so you can just see the on the bottom thing here, you can see where they started, and you can see how the pasture's regenerating as they move daily throughout the experiment. So in terms of the study areas, we kind of really expanded this based on people who were being involved, but also on the questions I would get from, oh, did you sample X, Y, or Z in that previous study? And when I was like, oh, no, I didn't, maybe I can integrate in that future. And so this is what I did. So we did for performance, we looked at weight gain, feed usage, FCR, and we looked at mortalities. At the site, we had a dedicated weather station, so we could look at site-specific temperature, humidity, and precipitation throughout the growth period. For the food safety, we looked at Salmonella, Campylobacter, E. coli, and the microbiome. We looked at environmental reservoirs, so in terms, we looked at the feed, the water. We looked at the flies using fly traps and wild birds using perches and looking at their feces. Excuse me. And then for the broilers, we looked at the feces that was on the pasture, deposited on the pasture the day before we sampled. We looked at the fresh feces, the cecal contents, the ileal contents, and the jejunal contents. In terms of physiology, we looked at gene expression, or going to look at gene expression and metabolomics at things in the blood, the cecal tonsils, mucosal scrapings in the liver and the kidneys. In terms of behavior and welfare, so I eventually linked welfare into this talk, near the end. But that's with Dr. Regmi, and so he put in some video cameras. So at different time points throughout the grow out, he did, I believe it was 48-hour recordings to look at the behaviors differences between the different breeds within the coops. And he also did some leg bone analysis that you'll have to speak to him more about. He knows more than I do. And then we did have the opportunity, because within our building, we have some meat quality people. So we did some meat quality analysis, looked at carcass rate, breast yield. You can see them there, but as well as we looked at myopathies such as woody breast, spaghetti muscle, white striping, things that are important in the industry, and to see what effect the breed had on those things. In terms of just some preliminary data, and this is very preliminary, in terms of the salmonella, if we look at the environmental reservoirs, the feed, we never found it. The water, it peaked very low. One sample at the very last sampling day for the slow-growing flock had a positive. But for the most part, they didn't have it. One thing we did find interesting is for the insects, once we were able to put the fly traps out, we consistently found salmonella within the insects, mostly they're flies, but they could be other flying insects in there as well. So that seems to be a potentially important reservoir. We'll have to do more work to figure out. And one time we found it in the wild bird feces. In terms of the bird itself, so this is the left one is the feces, the fresh feces from the large intestine during the necropsy, and the right one is the one that was deposited on fecal deposits on the pasture. But what you can see is in the orange, the feces from the fast-growing breed always was more. Usually more often we would find it in the fast-growing breed than the slow-growing breed. Once again, here's in the Sika. It eventually came up, and again, the fast-growing, the Cornish cross had more salmonella than the Freedom Ranger. And the Ilia, same thing. You see orange and not blue. The weird thing was for the final carcass rinse, we only found it in the slow-growing, but not in the fast-growing one. It was a flip. Not really sure what that means yet. We have some theories, but I can't really speak much more to that at this point until we do some more work. For some Campylobacter, what we found was that the wild bird feces and the feed was always negative throughout. But again, the flying insects, we were able to find some campy. So again, a potential important reservoir, which is something a farmer can do something about by just putting up fly traps. And what you'll see again is we were able to find it. Once we found it, we were able to find it in both. But again, you'll always see that the orange bar is higher than the blue bar. So again, the Cornish cross seemed to have more of the pathogen in it than the Freedom Ranger. And luckily, the final carcass rinse made more sense this time. Again, it followed the trends we saw up to that point. You know, it was higher in the Cornish cross and lower in the other one. So in terms of future goals, what we're doing this year is we're going to expand on that. Obviously, we have lots of data analysis to do on those samples, but we're going to continue to move forward with these field trials setup that we've done. So in the spring, summer of 25, we're going to work with the Freedom Rangers first. Excuse me. They're going to be hatched and brooded at UGA for a little bit more control. Then about day 18, we're going to look at the effect that raising it in a more conventional house setting versus in these pasture raised coop settings will have. So we'll have 4 coops out on pasture with about 120 chicks per coop, and then have 4 units, in-house units that are more reminiscent of a conventional system and the equivalent density in-house. And then we're going to raise them to 77 days. And we'll do the same sampling strategies and targets that we did in 2024. And then at the second part of the year, we will do the same study, but do the Cornish crosses rather than the Freedom Rangers, and see what effect housing has on that. And what effect do the access to the outside, the environmental reservoirs that are there, does it affect not only the food safety, but things like performance, welfare, um, uh, meat quality, all of those things that we looked at this time, we're going to continue to look at. Just try to get a more systems-based approach, because for a lot of those modeling analysis that I showed in the last study, that takes not only a lot of data to make them say something of importance, but the data has to be connected to each other. We can't just take data from one study and data from another study and then data from a third study and combine it together. Well, you can do it, but without linking the data directly together, the interpretation becomes much more difficult. And so we're just hoping to collect lots of data and see how all these things link up to try to get some ideas of, you know, how these different systems, you know, how it affects different aspects of poultry management, and, you know, ultimately the product that the consumer gets. Because, I mean, you want a nice, safe, clean product for your customer. And so hopefully some of this data will translate to that. So I'm just finishing up. I'd just like to acknowledge all the people involved here. The people in bold and underline are ones that were directly related to some of the data that I showed here, but everyone had played an important role in this work. As you can imagine, it takes a lot of people and a lot of hours to collect all this data. And so I'm thankful to each one of them for all of their help. And at this point, I'm willing to take any questions or snide remarks or whatever you feel like. I'm here to answer it, whatever you got.
Those snide remarks will have to go through me, so that'll be
Okay,
interesting.
that's fine. Do you want me to stop sharing my screen or leave it up?
You can
Or
leave it up for now.
okay.
Yeah, so thank you very much. That was very interesting. There are some questions from the audience, and we also have some questions. The first question was, I think, related to one of the earlier farms where you shared
Okay.
results from. The question is, how variable were the grass types in the pasture?
Uh, I would love to have an answer for that. That's not one thing we specifically looked at, honestly. I would guess there is some differences because, I mean, we had a couple farms that were in similar geographic regions, but even within, you know, some could be a couple hours away, but they'd be at least, excuse me. And where I'm in Georgia, you know, if I go an hour north, I'm up in the mountains versus if I go, you know, an hour west or even south, then it's a much different. So yes, there's variation. What that is, I can't specifically say. One thing, you know, for the new studies that we're doing, those will be uniform because I mean it's all in the same area. So we will. That was kind of the advantage of doing translating to the system to reduce some of that variation. But no, was it variable? Yes. Can I tell you what that variation is? No. I could find it out potentially if I still have a good connection with all the farmers involved. So if they haven't significantly changed what they've done since 2014 to 2017, or seeded it with anything different, I may be able to find that out, but I don't know it offhand.
That's okay. Then another attendee asked, what number distinguishes the difference between a commercial flock and a hobby flock? So I guess that's more of a
That was, well, I
general
mean,
thing.
what we did it, if I want to say, if I remember correctly, the cutoff was either two or three. I think it was one or two. If they did one or two flocks in a year, we
Mm,
considered it a hobby farm. Three or more, it was considered more professional. I
mm,
mean, that's the thing. But I mean, it was also obvious on the flock number, because usually the ones that did one to two flocks a year, they were doing somewhere 100 or less birds per flock.
mhm,
Whereas the ones that were doing more, I mean, they would do 300 to 500 to 800 per flock. So, it was
right.
also a flock number as well, but, we just did it by some flock size. Well, at least that's how we defined it. I'm not saying that's a universal definition,
Mm.
but in terms of that paper and that analysis, that's how we defined it.
Great, thank you. Another question is, how do pastured poultry Salmonella serotypes differ from the serotypes in conventional farming practices in regards to human health hazard?
I mean, I would say they looked. I know, like for instance, Kentucky is not a human health hazard, but it's in the U.S. That's by far the most prevalent one in commercial
Mhm.
settings, and that was by far what we found most often in the pastured poultry. So that was good. I would say, you know, I know now. Again, this happened. I don't know how many years ago. It doesn't seem that long ago, but apparently it's like 7 years ago we did this. Um, we have seen some shifts in the commercial setting of some serotypes increasing. So, for instance, Infantis has really popped up as being a more important Theratype probably since 2017, 2018. And we did not find that very often in mine. But again, mine stopped in 2017. I will say in 2017, I did start seeing more Infantis than before. So it seemed to be that's like a universal poultry problem. Now it's probably exacerbated much more in a commercial setting because you shove 20,000 of anything in a small area, something gets sick. I mean, if anyone here has kids in an elementary school, there you go. One kid gets sick, everyone gets sick. And so, that doesn't help the problem. But I mean, I did not see a large prevalence of the like the most notorious serotypes that you'll hear for human health. So, I would say at worst, it's on par with commercial and potentially, but considering the fact that it does have so many different environmental exposures than a conventional would, I mean, theoretically, you would think, well, if it has a lot more opportunities to pick up various serotypes from different sources. It didn't seem to be the case. But again, that's just within that data set there. But I was not thinking like I did not see them be like, oh, it's much, there's a much higher risk from commercial. No, and I didn't see. I will say I did see some serotypes I'd never heard of before. So it did definitely have unique serotypes, but they weren't ones known to cause human illness. If you're specifically wondering about those serotypes.
And I know you've been talking about salmonella, but I think the general thought for pastured poultry is sort of the general sort of idea there is that they are at risk of more pathogen transmission. Could you comment on that? Is that
I mean,
accurate?
I
Yeah.
can, I can, I can see it going one of two very different. It's two different schools of thought in my mind. One, like you said, you're being exposed to, all these different insects, all these different wild animals, environmental. You're outside, you're near another. Most of these are in rural settings. There's other uh, uh, agricultural settings that you can get transferred through the water, through the wind. So yes, you have a lot more exposure, higher risk. But alternatively, I could also think it's like, when it's as long as all of these uh, contacts are relatively low concentration, it could also be training their immune systems better than something in a house. And so if you're trained at small levels, you're kind of, I won't say immunizing, but you're testing their immune system against smaller doses all the time of all these different ones. So in the long run, it may make them somewhat more resilient, potentially. I'm not saying it does. I could just see the argument for that. So I could see it going either way. Now, broilers, they don't live very long. Now I could see, especially like in a layer, I could see that being much more of a, because they're, a year and a half, 2 years in age, you could see that resilience build up. Whereas in a broiler, you're kind of out the door within, 72, 77 days, and that was even long. So I could see either way. It really depends. I mean, I think one of the things we're seeing now is like with the whole avian influenza thing. That's probably an issue just because that's known to be wild birds. They're out in the environment. And if you have these pasture flocks out in the environment, there could be direct contact between the two. Um, but I don't think I mean, that's just an issue because the birds are in the environment. It's not because the management system is poorly designed or developed. It's just a fact that there's different species living in the same space and one's known to carry a disease that we can't really do much about. So um, I don't know. Now, I've seen nothing in all the data I've seen and collected that would tell me, it's significantly, more of a problem than in a commercial setting.
Mhm. Oh, that's very promising to hear.
Yeah.
Were flocks in your studies ever vaccinated with live salmonella vaccines?
No, the only thing every once in a while you have the option when you buy the chicks. So the few times, like I said, some of the farmers were on board with us controlling some more things. And the few times we purchased chicks, I believe we had them vaccinated, excuse me, against Merrick's disease, but that's it. No, there's no, there was never a salmonella vaccination in any way. No.
Could you comment on the difference of your processing whole carcass rinse outcomes and the final whole carcass rinse outcomes?
Yeah, that's well, the difficulty thing there is there's such a diversity among the ways that the processing was done. So I had some farms that did on-farm processing, usually the ones that were smaller, obviously. So they would do on-farm processing. They had small-scale stuffs, and some were much more organized than others and had better equipment than others. And then I did have the set of the professional ones. Mostly they sent them to USDA inspected small processing facilities. So you had all those variables in addition to what the final like. So the consumer, I would ask the farmers, okay, once you have processed, how long does it take to get that product to your customer? And what do you do to it in the interim? So it ranged from some were like a CSA type where they just told their community members, hey, I'm processing today. I will be done by 4 p.m. Come pick it up. And they would pick up just a chilled carcass, and that was it. Whereas others would be, they were in markets in different cities, and so they would be frozen for like a month. So I ranged anywhere in between, and I followed whatever that farmer did. I didn't make that uniform, so we did see a lot of differences. I will say, largely, one thing that we saw that would reduce it in the final product versus the processed whole carcass rinse was if it's frozen, it did tend to definitely drop everything. Freezing it, and the longer you froze it seemed to have a greater effect. So that's one recommendation. But again, I know for some of the smaller farmers, they don't have the freezer space to do that. And I recognize that. So I mean, it's not a universe, but if you, and I did eat some of them, I'm like, if you have the ability to, I would recommend that. Freeze it. That was one of my biggest things. But we are doing with some of the modeling of some of this data, we're still doing it. We have found that the use of organic acids, sprays, and dips in the processing did seem to reduce E. coli levels pretty significantly. And granted, the E. coli probably isn't a pathogen. It's just a general E. coli for just general contamination or general microbial quality estimates. But still, if you're knocking down an E. coli, most likely you're knocking down the others too. So, like an organic acid dip or spray somewhere in the processing, if able to, is a good idea, I would say. Those were just some of the things we noticed, general things we noticed.
Great. Another question was whether the chicks in the flocks were assessed for salmonella at placement, so the chick boxes or the paper.
So for the large study, the first study the big one I showed, no, we did not do that at that point. But for the ones going forward, yes, we are going forward. We are checking the chick papers, the chick boxes. The one that I showed that was done in the summer of 24. Uh, we bought those from, hmm, I want to say Murray McMurray hatchery. And so we got those. So we did test the chick pads from that to know whether what their status was initially. Problem with that is we don't have any control. We didn't have any control of when they were shipped to when they arrived. So I didn't have a true day zero, which we wanted a true day zero. So moving forward, we're going to actually get the eggs and we will hatch them at UGA. So we'll be able to swab the eggs and have like egg incubator swabs before and to really get an idea of what they are coming in before they even get anywhere, what their salmonella status is, and other pathogens.
Great. Which section of the intestine is most representative of the poultry microbiome, and can fecal samples serve as a reliable proxy to assess the bacterial community?
That's an excellent question. So it depends on what you're interested in. I mean, if you're interested in what you're going to be receiving on your plate when you're eating it, then probably none of the, well, we have data that shows none of that is very representative of that. Um, I would say generally the cecal and the fecal are close to one another, especially if it's fresh fecal. Now, for the pasture feces stuff that's been sitting up to a day on the pasture, that will be different because, I mean, you have different environmental effects of sitting out there, drying out. Other animals, insects, anything can step on it, move on it, transfer stuff to it. But the fresh feces versus the Sika are similar. The Sika's always been a little hard for me. I mean, I understand why it's used because you can find things food safety wise there. But again, it is so unlike any other part of the intestine because it's not an open-ended tube, it's a pouch. So things sit in there for longer, potentially longer periods of time and things like that. So I think it's interesting. The Sika is interesting to show the potential of what could be other places, but it may not be super representative. I would say like maybe the ilia is a little bit better than that, for what's going on in the bird itself. But again, we just did contents. I do know that Dr. Laura Ellistat, who does the physiology stuff on these projects that we're doing now, she prefers to do the scrapings rather than the contents. But again, she's looking on what's going on within the bird, whereas I'm kind of more interested in what's passing through the bird to the environment. So it really depends on what aspect of the poultry you're interested in to kind of figure out what the most appropriate sample to target would be.
Thank you. Final question.
Sure.
You had some really nice recommendations for professional and hobby farms to reduce salmonella prevalence. For a new producer setting up their outdoor access, what would you say are the top considerations for establishing their outdoor area to be sort of proactive in mitigating microbial issues in their flock?
So I would take a strong consideration into um, the topology of your farmland and how you're going to run your flock. Because ideally, you're not going to want, I mean, again, and this is dependent on your own, how much land you have, obviously. But if you have enough, you don't want to run, I wouldn't recommend running your coop over the same spot within the same flock. You want to give it some time to grow back up, the pasture to grow back up, regrow, normalize before it goes back over. Also, I've seen it often, and it's not, I mean, it can be a food safety thing, but it's more just an animal welfare health thing, is really be mindful of where the low spots are and the depression areas are on your thing. And if you can avoid them, great. If you can't, just make sure to know, maybe put some extra hay or something down if it's sitting there. Because if you get a rain event in a depression and then all your chicks are going to be stuck there and then they're going to start piling on each other. So one, you're going to have mortalities through that, but then also that'll cause a lot of stress. So if you have a lot of stress, the birds are going to start freaking out and probably defecating a lot more on each other. And so then you increase your likelihood of if there is anything that's getting transmitted between the birds a lot more. So keep that into consideration. Um, I think those would be two good ones. Kind of having a knowledge also of what's around you. And again, like it goes to topology, but like when we did the farm on this current study, the 2024, we saw it was relatively flat, but it was downhill of the neighboring farm, and that neighboring farm had cattle. So I was just interesting. I knew where, okay, let's make sure we don't cut down all the grass right near the edge of that fence. Have at least some kind of natural, try to filter everything because a good root system, a good vegetative barrier between the farms in order to, will it be foolproof? No, but kind of things like that, kind of getting awareness. You can do a lot of stuff like that. And also, if you have ponds and stuff on your place, that's where wild birds and wild animals will congregate. Maybe don't have your birds be able to access those physically to reduce any potential effects of outside reservoirs. And based on this last year, I would say fly traps, put fly traps up. I was surprised. I didn't think it would be as prevalent as it was, but it was found all the time. Now we still have to do more analysis to see what serotypes and what those types of salmonella and campylobacter look like versus what we found in the bird itself. But anything you can do to reduce it, and that's a relatively simple thing to put up to reduce it, and just to empty it every once in a while. And I have seen people do things where they put up, they're not chemical based ones, I forget what they were, but they basically would trap the flies and then basically collect them, and then since they weren't chemical based, they would just kind of put that within the feed and to give another protein source within the feed for the chicks to eat. So it was kind of a value added kind of thing. You're keeping it away. Maybe you can do something with it to treat them, even like compost them, whatnot, and then feed it back in, somehow, to recover it. But, all things I would think about, as someone starting up new, and start low numbers, get used to it, and expand as you're able to, and as you feel more comfortable.
Great. Thank you so much.